Hi, everyone. It is my pleasure to introduce my dear colleague and close collaborator, Dr. Snyder. He's an associate professor of radiology and imaging science at Indiana University School of Medicine. And Dr. Snyder, he has a strong professional background in ligand and the radioactive tracer development. He received his PhD in medical chemistry from Purdue University, developing novel dopamine receptor. Later, he joined the nuclear medicine division of University of Michigan Medical School as a postdoc researcher initially, and later become an associate professor. And he was trained there in PET radiochemistry and imaging. He remained at Michigan for 14 years before he joined and recruited by St. Jude Children's Research Hospital to design and direct their new molecular imaging research laboratories. And fortunately, in 2019, we have the pleasure to have Dr. Snyder join us and direct our molecular imaging ligand development program. This program comprises both IVIC Radiochemistry Facility and independent research programs. His team routinely prepares 6 or more different non-PET tracers for clinical and preclinical researches. And he also supports multiple collaborative study and projects to develop a novel radio tracer for imaging and for PET. So now we can hear more about his exciting program. Scott, please take the floor.
All right. Thank you, Yu-Chen. I appreciate it. And thanks everybody for joining this morning. It's good to see so many participants. Let me share my screen and put this thing in presenter mode. So as Yu-Chen mentioned, I've been doing radiotracer development research for about 30 years now, actually. First at Michigan and then St. Jude and now here. And over the course of this, we've done a lot of things in collaboration with either clinical or basic science research investigators to help to facilitate and promote their translational research. So what I'm going to try to share with you today is a little bit about what we do, and then some examples, in the second half of the talk of what we might be able to help, individual research projects with. So as Yu-Chen said, I'm the director of the Molecular Imaging Ligand Development Program. And this encompasses both the Radiochemistry Core, that's part of the In-Vivo Imaging Core, and individual collaborative research projects from 4 different radiochemistry faculty members. We have a cyclotron facility that enables the on-site production of short half-life radioisotopes. We can also work with generator produced nuclides such as gallium-68, which is used a lot in the clinic. And then we also have access to external suppliers for longer half-life radioisotopes that you're able to actually order and get shipped here in time. My team at BRTC is 3 staff scientists and Dr. Min Wong, who's a research faculty member, and then my collaborators also in radiochemistry, Mark Green, Mike Schulte, and Chiwang Cheng. Would be, we do a lot of collaborative projects, both amongst ourselves and also with external folks to get projects off the ground. So here we are out in BRTC, where the Cyclotron Radiochemistry Facility is. The In-Vivo Imaging Core imaging facilities, some of them are in Goodman Hall. There's both human and animal scanners in Goodman Hall that we ship radiopharmaceuticals to frequently. And then the remainder of the research imaging is in the R2 building on Walnut Street. So all of the radioisotopes we produce out in BRTC either go through a pneumatic tube that runs underneath the ground parallel to Indiana Avenue, underneath the river, and ends at R2. And we can basically shoot a dose of one of our radiopharmaceuticals in about 90 seconds from BRTC to the R2 building. When we have to send something to Goodman Hall, those go by regular courier. So we have a courier that we work with that will transport radioactive material up 16th Street and over to the Goodman Hall building. So our main focus is the development of radiopharmaceuticals to investigate disease, primarily with PET imaging. And I'll talk in a minute about specifically what that is. But we provide established radiotracers to the community, things that have been published. There's a bunch of things that we already have established here that we make all the time that we can supply. We do radiochemical development, so novel radiotracers. If somebody has a new lead compound that they're interested in getting some in vivo biodistribution and kinetics on, we're happy to talk about those kind of projects. Those ones are done usually more as a collaborative project, but we do all that. And we help with all of the regulatory documentation. So the animal approvals, the Radioactive Drug Research Committee or FDA submissions that you would need to do this kind of work in living subjects. So what is a radiotracer? Basically, a radiotracer or a radiopharmaceutical is some sort of biologically active molecule that you've tagged with a small amount of radioactivity. You inject that into your test subject, whether it's an animal or a patient, and you use a special camera to follow the radioactivity as it travels through the patient's body. By measuring the location and residence time of that radioactivity, you can make specific biochemical measurements of active processes in the body. We can do things like measure blood flow. We can measure enzyme activities. We can measure receptor occupancy rates, things like that. So you can get actual quantitative data out of these images. When we're developing a new radiopharmaceutical, the first thing we need to know is, what target are we looking at? Are there molecules that are already published or available that we can use as a starting point to do the radio labeling? We have to develop the actual radiochemistry and purification systems, quality control systems for that. We validate the new radiotracer, usually starting in vitro systems, but then moving into animals to make sure that we're actually imaging and measuring what we think we are. And then we develop the more elaborate imaging methods for specific animal models or patient groups. There's basically three different ways, the way I think of it, there's three different ways that you can use a radiopharmaceutical. You can do direct labeling of drug candidates or specific biomarkers. You can use radiotracers that measure a biomarker relevant to your disease, but not necessarily the drug that you're looking at. So there are biomarkers, for instance, specifically for neuroblastoma tumors. And you can see, is your tumor growing or decreasing in size based on whatever it is, the therapy that you're looking at. And you can do some basic metabolic imaging, things like, again, like blood flow, glucose metabolic rate, and see how those are changed in the course of the disease or as an effect of some therapeutic. As I mentioned, there's a couple different ways of imaging these radiopharmaceuticals. First, there's what's called SPECT imaging, or single photon emission computed tomography. This relies on a radioisotope that emits a single photon of energy every time one of the radioactive nuclide decay. It's a planar detector that rotates around the patient and measures these decay events as it goes around. And it's collimated so that it only is measuring things that are coming in perpendicular to the scanner. So it's relatively certain of where the radioactive decay event happened. These cameras are really widespread. I mean, almost every hospital has a SPECT or a gamma camera in it. So it's easy to get access to. There are a number of commercially available radionuclides and radio labeling kits that you can use for these. So this is the predominant nuclear medicine modality in the clinics. Lots of things you can use with a generator that you don't require having a cyclotron on site. PET imaging, or positron emission tomography, relies on a different type of decay that results in two equal and opposite gamma photons being released from the body that allows us to sort of triangulate where this radioactive event occurs. So we get these what's called coincident detectors that detect the photons on either side of the event at the same time. Because we've got a ring of detectors, it increases the sensitivity or detection efficiency of these cameras by about a factor of 100 over the SPECT camera. Because we can do this triangulation, we can actually get quantitative images out of it and much better resolution, giving you about an overall 2 times sharper image with the PET. The downside is it's more expensive, and for many things, you need to have a cyclotron facility, at least within the immediate area, if not on location. So what are some of the typical PET radioisotopes? So these are just 4 that we can make here at the BRTC facility. As you can see, the most common PET radionuclides have a very short radioactive half-life. This means that for carbon-11, for instance, I'm losing half of my material every 20 minutes. So this folds into how we do the chemistry. It folds into limitations in transport. We have a study with O15 water. We've only got a 2-minute half-life. We can't do that at Goodman Hall because we don't have time to drive it over there. That has to be done in R2, so we can use the pneumatic tube system to send it. These isotopes are produced in a cyclotron. This is the Eclipse that we have here in the basement of BRTC that we share with PET Pharmaceuticals. PETNet makes all of the FDA-approved drugs that have to be done under strict GMP compliance. Our lab does all of the research radiotracers that could be produced under slightly relaxed regulatory environment. Once we get our radioisotope, that's transferred to the radiochemistry lab, and we do all of our radiochemistry remotely inside these lead-shielded fume hoods. Again, all the PET isotopes give off 511 keV gamma radiation. So this isn't something that we can do on the benchtop. This has to be done behind a couple of inches of lead. So all of the chemistry is done with these automated modules that reside inside what we call hot cells, these lead-lined fume hoods. The modules themselves are a series of valves and switches that allow us to move reagents from reservoirs into our reactor vials. We can do heating and cooling. We can do HPLC chromatography purification inside here. So starting with, for instance, carbon-11 CO2, we can make carbon-11 raclopride using this setup and have it be pure enough and in a formulation which we can inject into a patient. All this because we can't touch anything. This door that you see here with the window in it goes up, and everything is controlled by a PC that's on the outside of the hot cell, so that the operator never goes inside. There's a number of different these radiochemistry synthesis modules, as we call them. The initial ones are similar to the ones I've shown here on the left. These are sort of the homemade versions. Everything is fixed reactors and fixed tubing, fixed plumbing. This is the one we have at BRTC. This is one from one of my former institutions. These are very flexible. They're great for doing radiochemistry research in, but the downside is every time you do a synthesis in here, particularly if you're doing it for patient, this entire module needs to be cleaned and sterilized before you start to work, and all your reagents need to be filled up and topped off. And it's about depending on the tracer, it's between 45 minutes to an hour and a half setup before you even get to start doing anything with radioactivity. So around the year 2000 or a little bit before, some of the commercial vendors started to make a little bit more streamlined modules. So this is one of the initial ones happens to be from GE. I'm not trying to promote their products, but this is another. It's still a fixed reactor module. Still has these reservoirs of reagents, and still has the tubing and valves that need to be cleaned up in between times. But it's a commercial project with a preventative maintenance contract that you don't have to repair and upgrade, yourself constantly, so it doesn't become obsolete. Because all this cleaning is kind of a pain, and because the FDA likes things that are single use, eventually the field moved to more of these kit-based or cassette-based modules. These ones have a single-use removable flow path, and all the valves are these little plastic valve heads that are disposable essentially. So when you use these kind of modules, the flow path gets put together for a single use. You use it, you pull it off, and you throw it away. They're all barcoded so that you could do inventory control on them, and we know we haven't used a cassette before. Most of the more modern modules are moving in this direction. In fact, some of them have gone to the point where for very routine radiopharmaceuticals like fluorodeoxyglucose, FDG, that is used in every nuclear medicine clinic every day, they've developed a really streamlined synthesis for that allows it to be done on one of these much smaller modules with a much smaller cassette. In this case of the IBA Synthera module, the cassette is literally the size of a pen. So these are, again, single-use cassettes, much lower footprint, not as flexible for doing research with, but very good for doing daily routine production. Thanks to some recent funding from CTSI Core Equipment Grant, thank you very much. We were able to purchase one of these cassette-based systems, the GE Fast Lab 2, that we just installed literally last month in the lab. So we're working on getting that up and working and implementing some new radiopharmaceuticals on that. And this will support expansion of our program so that we can do more things in a day and serve more of our collaborators and customers. This is just a scattering of some of the radiotracers that we are capable of making. These ones are ones that we kind of make more often than some others. Things as simple as O15 water or C11 acetate, or something more complex like fluorine 18 phthalate for dopamine receptor imaging. We make routinely for either clinical research or animal studies. In the last couple of years, I mean, you can see this is the sort of volume we do. It's not tremendous compared to some of the really big radiochemistry labs, but we do ship a lot of radiopharmaceuticals out of here on a monthly and annual basis. Just as an example of some of the things that you can do, you know, we make fluorotyrosine for brain tumor imaging routinely for the clinic. C11 acetate and palmitate are used for lipid biosynthesis. Acetate is more for oxidative metabolism. Palmitate is more for, you know, lipid synthesis. PIB is a beta amyloid tracer that's used in Alzheimer's disease imaging. Both raclopride and phthalate are dopamine D2 receptor agonists. Glutamine metabolism with FSPG, and of course water I mentioned before. So as you can see, there's pretty much any biological process that you can imagine. There either is or there could be a radiotracer to measure that process. We're currently in the implementation phase of 4 different radiopharmaceuticals right now. The UCBJ C11 is something that Dr. Wu and I are working on, and we have another investigator who's planning to submit an R01 application using this sometime either this round or the next round for NIH. We have some requests from the clinic for parathyroid imaging with fluorocholine. So that one we're getting going. MFBG is a norepinephrine transporter tracer that is used for both cardiac imaging and neuroendocrine tumors. And I've mentioned the glutamate metabolism with FSPG. These 3 fluorine 18 radiotracers are literally being enabled by this recent purchase we made with CTSI money of the new cassette chemistry module. So we're actively, I divided my group into 3 little, you know, sub teams to implement all 3 of these radiotracers essentially at the same time. So we'll see how that goes over the next couple of months. Dr. Mark Green over in Goodman Hall has another radiochemistry lab that does mostly radiometal chemistry. So he works a lot with gallium-68 and copper-64. The gallium is a generated produced isotope. The copper we order usually from Washington University. He's got 2 active radiotracers that he makes on a sort of weekly, monthly basis, and 2 more oncology tracers that he's in the process of getting validated, so that we can get those into studies as well. Most of what he does is the biomolecule labeling. I'll talk a little bit about that later in one of the projects that I'm doing, but that's his forte. So if anybody wants to do, you know, peptide labeling with gallium, he's the guy to talk to about it. So how does PET actually work? All of the PET radionuclides are 1 neutron short being stable. So in the case of carbon, carbon-11 has 6 protons and only 5 neutrons. Stable carbon has 6 of each. Because in my non-physicist view, because you're short on neutrons, your positive charges in the nucleus are kind of too close together for comfort. So this becomes an unstable nucleus, and it has to get rid of all that excess energy somehow. And the way it does it is by converting one of its protons into a neutron by ejecting a particle called a positron. This particle has the mass of an electron, but is positively charged. So essentially, it's an antimatter electron. This positron will travel a very short distance in the surrounding tissue until it encounters another normal electron in the area. And for those physicists and Star Trek fans in the audience, what happens when identical particles of matter and antimatter collide? They explode. And all of your mass gets turned into, or most of your mass gets turned into energy in the form of these equal and opposite 511 kiloelectron volt gamma rays. And these are the gamma rays that we use for that coincidence detection in the scanners. So this is my little cartoon version of a PET scanner with my little patient's head inside here, and they have maybe a glioblastoma in their head, so they've got this high concentration of radioactivity that's been taken into that tumor. The scanner is recording decay events only when the detector on opposite sides of the ring are triggered at the same time. So in that way, all the scattered photons don't get recorded as actual decay events, and you generate essentially a radioactivity concentration map, a 3D map of the radioactivity concentration in the patient. The picture on the right is a typical PET scanner with the gantry that the patient goes through and the aperture the patient goes through and the gantry that they lay on. We have small animal versions of these. So this is the PET MR that's in the basement of Goodman Hall. So this is a 9.4 Tesla MR magnet that Dr. Wu's group can do all sorts of fancy stuff that I don't understand, to do molecular imaging on the non-radioactive side. And then this little PET detector insert can be pushed right into the bore of the magnet and give you simultaneous PET and MR imaging. So this allows you to get the quantitative biological process measurement from the PET with the anatomical and some flow measurements that you can get from the MR. So these things are getting more and more prevalent and hopefully more and more useful. I know they're being used much more in our facility as every month goes along. So I mentioned there's a few different ways that you can use radiopharmaceuticals for to support translational research. So for the rest of my talk, I'm going to try to lay out just a few examples of those things that either I've done or we do here at our facility. But let me take a second and see if anybody has any questions on anything so far. Trying not to go too fast, but I want to make sure I don't run over time. Okay. So my first option for doing radiotracer research is the direct labeling of a biomarker or a drug candidate. By actually labeling your drug, you can get direct in vivo biodistribution, kinetics, and metabolism information, and usually make actual quantitative numbers out of that. You can easily demonstrate target engagement. Does my tracer get where I think it's going to go? Does my drug get where I think it's going to go? You can measure occupancy, how much of my drug am I actually getting to where it's supposed to go and how much is ending up in the liver. You can get some information on target selectivity because I can do a whole body image on a mouse and see where else in the body my drug is going besides where I think it's supposed to go. The downside of this particular method, though, is that every time you have a new project, you have to have a new radiotracer, which means all the upfront development costs on getting something labeled and validated, which on my groups, is not days but like months of effort, and it gets kind of pricey after a while too, because you're paying for cyclotron time, you're paying for tech time to get this work done. You can get a lot of really good information out of it if you've got a drug candidate that you think this is the one. I just need to get some in vivo data to convince the FDA that it really is the one. This is the way to go. Usually we do these as collaborations just because it would be prohibitively expensive just to pay the core to do this kind of work. We're talking like a new radiotracer ground up development to the point of being in patients is about $100,000. So one example of a project I worked on at St. Jude related to this, we were doing a collaboration with Tom Webb, who was then in the chemical biology department at St. Jude, is now has moved on to industry. But he was looking at these pseudomycin compounds, as he dubbed them, that are they're alternative splicing modifiers. A lot of tumors have alternative splicing of peptides, so of their mRNA that give aberrant peptides. So he thought that these would be a good drug candidate, and they would look like they were showing some promise in like cell toxicity studies. So we wanted to do some in vivo studying with them and see what their biodistribution was, and in particular, he was interested in the brain penetration of these radiotracers. So I'm going to show you a short video here, and this is a mouse just to orient you. This is a highly overexposed CT image of a mouse, so you can see the whole mouse's body and the nose cone he's laying in and everything. This wire-looking thing that's going here is actually a jugular catheter that's going into the mouse. So what you'll see when I click the O button here is you'll see actually just the skeleton of the mouse at that point. And you'll see the radioactivity as a rainbow color coming in, with the reds being the higher amount of radioactivity and the blues being lower. You'll see the activity flowing in through the catheter, sort of perfusing through the cardiopulmonary area. Eventually, you'll see it kind of draining into the kidneys and into the bladder. And most of the radioactivity you'll see kind of ends up in the bowel. So let me start the video and see what that's doing. So there's the mouse's skeleton. There's the activity coming in through the heart, the lungs. Now it's coming up into the kidneys. You see the kidneys right here. As it clears through the kidneys, you'll start to see a spot down here where it's draining into the bladder. And then what doesn't get cleared through the bladder ends up in the liver and the gut. So all this stuff that's here is most of it is GI activity. So let me try to play that one more time, see if I can get to do it. And I want everybody to pay attention to the rat's head when we do this one. So let's see if I can get him to go again. There he goes. Okay. So what's right up here where the head is? So here's where I want to know, do I have brain penetration? So the rat's brain is right here where my cursor is. And as you can see, there is absolutely no penetration of this radiotracer into the brain. So there's no penetration of this drug into the brain either. So whether that was a good thing or a bad thing for him doesn't matter. We gave him the answer that he was looking for in terms of his FDA filing, so he can tell them how much of this drug is potential to get into the brain. So those are the kind of kinetic data that you can get out of these, and you can, like I said, make quantitative numbers out of those images eventually. That was a carbon-11 labeled radiotracer. We've also done a lot of things with fluorine-18. People don't really think about this, but there's a lot of approved pharmaceuticals that contain fluorine, and many of them, the vast majority of them, can be labeled with fluorine-18 without modifying the drug at all. So there's a lot of things you can do with direct labeling of drug candidates with fluorine-18. Worst case scenario, you can add a fluoroethanol someplace, or you can add an ethanol or add a fluorine onto an aromatic ring someplace that doesn't too adversely affect your biological activity, but will give you the tag to then do the labeling. So just an example, here's 4 known drugs that are on the market. This is Plavix, this is Flonase. So these are things that people use every day that have fluorines in them that could potentially be labeled. This is a direct labeling project that I've been working on in my lab for a few years now. So I'll just take a couple of minutes to show you this. This involves antibodies for the diacylganglioside GD2. So this is a glycosphingolipid that's expressed in the surface of a number of different neuronal cells, particularly paraspinal neurons, but it also is expressed on quite an array of different cancers. In particular, neuroblastoma and melanoma cells are almost universally GD2 positive on them, and they have quite a bit of GD2 on the cell surface. As a result of this, one of the anti-GD2 antibodies that's been developed over the years is dantuximab, which is now marketed as Unituxin, was FDA approved back in 2015 for recurrent neuroblastoma, and showed some really impressive improvement in patients' overall survival. And 20%, 10% might not look impressive, but these are patients that currently have a 40% survival rate. So if you're going from 40% to 60%, it's important for the patient. So these are very promising chemotherapeutics. The thing with them is, though, not all of those tumor lines have this uniform high expression of GD2. And in fact, in the literature, it's reported that greater than 88% of osteosarcomas are GD2 positive based on immunohistochemistry. However, there's been 2 different clinical trials, both of which failed because this drug had no effect, where it's having this really nice effect in the neuroblastoma. So why is that? And our contention is that if we could make a PET radiotracer based on this therapeutic antibody, it would first of all give us in vivo biodistribution, target engagement, maybe dose titration information, because we're directly labeling the drug that we're given. Second of all, we can use this for patient selection, because if my radiotracer doesn't bind to the patient's tumor, then neither is the therapeutic antibody going to bind to the patient's tumor. And ultimately, what we'd like to do is label this antibody with a therapeutic radioisotope, so something that will actually do what's called radioimmunotherapy. And the way that works is all biomolecules, or most biomolecules, when you label them, they have a longer half-life than most small molecules. So most drugs, a lot of drugs have, sort of minutes to hours, maybe days half-life at the most. The average biological half-life of an antibody is 2 days. So we can't label this with carbon-11 and expect to get anything useful out of it, because we'll still have all the unbound antibody floating around in the patient. that'll confound the imaging. So what we do is we label those with isotopes that have a much longer radioactive half-life that matches the biological half-life, and that means basically using radioactive metals. So you take your peptide or your antibody, you put a chemical linker on that and link it to one of these polyvalent molecules that will bind or chelate metals. And you can use the right chelating molecule that will be selective for a particular radioactive metal that you can use. So copper versus zirconium versus gallium versus yttrium, whatever you want to use. So we took some of another one of the anti-GD2 antibodies. This is the HE1418K, 322K. Don't worry about the name of it. This is another version of dinutuximab, essentially. Sorry, second generation, basically. And we labeled that with both copper-64, which has a 13-hour half-life, and zirconium-89, which has a 78-hour half-life. So the thought here is, if we've got a relatively short, so 13-hour half-life radioisotope, we can do serial imaging on some of our model animals and image them like once a week if we want to. And the radioactivity will have decayed in between times, so it won't mess up our images. If we're going to do patient studies, we might need something that's going to be a little bit longer, so we give our antibody at least a couple of biological half-lives, so say like 4 or 5 days, for the unbound stuff to clear out and give us cleaner zirconium images. So this is what the images look like. Again, this is a PET CT of a mouse. This one has bilateral flank tumor. So this is a GD2 positive HLA-20 tumor. This is a GD2 negative tumor on this line. All the stuff in here is the background that I was talking about. Copper has the added problem that there's a lot of copper binding proteins in the body, so it does what's called transchelation. Basically, the copper falls out of our radiotracer and binds to things that are already in the body. So it gives you a little bit of background, but you can still definitely see the tumors without any problem in this. If you take the same antibody and label it with zirconium instead of copper, this is what you get. Zirconium is essentially bioorthogonal. It has no use in the body, so if it falls out of the antibody, it just clears to the urine. There's very little background in these animals with the zirconium. Again, the animal's flipped over, but this is the GD2 positive tumor, this is GD2 negative at 2 days after the same time point after injection of the radiotracer. If we wait a little bit longer for that biological clearance, now we've got the GD2 positive tumor showing up and pretty much nothing else. So these were some, to me, were some pretty amazing images. We were getting, 6 and 7 to 1 tumor to background ratios, which is really good for most tumor imaging agents, particularly in rodents, because they tend to have a pretty high background. So this was part of the answer. We see the biodistribution, we can see the intake and the clearance kinetics on it. We did an experiment where we actually gave our radiotracer with a therapeutic, so the 16 milligrams per kilogram is the therapeutic dose of this antibody, and then about a quarter of that dose, and then looked at the biodistribution of those, so we could tell, where this radiotracer was binding. And the way this works in antibodies is, if you give the non-radioactive version with the radiotracer, the bulk of the mass of the unlabeled antibody will basically saturate some of the low affinity, high capacity binding sites that are mostly the metabolic sites, and things like fat and that just take up radiotracer and give you background. So you don't see much change in those areas, but what you do see is an increased availability of the actual radiotracer to bind to the tumor. So our tumor numbers went up with the dose of cold antibody co-injected with our radiotracer. The other interesting thing that I thought was the bone numbers also went up. This would seem to indicate actually specific binding in the bone, but I think mostly what it is, I don't think it's marrow. I think what it is actually more available zirconium to bind to bone, which it does do in rats. For some reason, rat bone binds zirconium, but human bone doesn't. The other thing we got out of this is, by doing the math, we could tell exactly how much of our drug dose was getting into the tumor. So at a therapeutic dose, or even a lower dose, we're getting about 20 to 25% of the actual dose of the drug is getting to the tumor. And we made these measurements using the image. Our second question was, can we use this to stratify patients, right? So these are 2 osteosarcoma xenografts, patient-derived xenografts. The one on the left, I'll show you the flow cytometry on it. The one on the left is basically 100% GD2 positive, and it's Fluorescence intensity was pretty high. So all the cells are GD2 positive. All the positive cells have a lot of GD2. The other cell line was about 70% GD2 positive, and the actual expression per cell wasn't as high. And we can see this in the images. You can see a much hotter uptake of the radiotracer in the higher expressing tumor than the other one. The really interesting thing to me in this is that the way they're currently stratifying patients is using immunohistochemistry staining of biopsy samples. So here's the immunohistochemistry staining of these two cell lines. This is the GD2 staining of the highly expressing and the lower expressing xenograft tissue slices. And I'm not an immunologist, but I don't see any difference between these two. So in these clinical trials, the clinicians aren't really effectively stratifying the patients. They're giving the therapy to a patient who they could say from the outset it's not going to work on. So we're really excited to try to get this into the clinic and get it into helping expand the use of this antibody and help some of these clinical trials. So the second way that you can use radiotracers to advance your research projects is sort of the more common way to do it. These are the indirect measurements of some biomarker for disease or biomarker for your effect. So what I mean by this is, again, if I'm looking at a therapy for neuroblastoma, I don't necessarily need to label my therapy. I just need to label the neuroblastoma and see how that changes over time. So we can use, in many cases, known radiotracers, published radiotracers for a therapeutic target. Then we can still get target engagement. You can still sometimes measure occupancy if your tracer is binding to the same place that your drug is. That's not always the case, but you sometimes can get that information out of it. You definitely can measure the progression of the disease or the therapeutic effect if you're having it. This can either be established radiotracers like the things that we already have that we can make currently, or these would be published radiotracers that would be relatively simple and less costly to implement in our facility for an ongoing study. So we have a request to implement the fluorocholine. It's a known radiotracer. There's dozens of chemistry papers about it. It's something that we can do reasonably quickly and reasonably inexpensively. And this is basically the type of thing that we do as part of the IDSC core resources, sometimes mixed with collaborative efforts, depending on if, it's like the precursor molecules are commercially available. We do a lot of projects where we're collaborating with a chemistry group that either is doing drug design or is just working with us to do the organic chemistry to make the labeling precursors, all of the non-labeled standards that we need for the analysis. And then we work out the radiochemistry and biological end of it. These collaborations have really worked well for me over the years. The radiochemistry labs typically don't have the capacity to do full-blown, med chem studies on something to design a new radiotracer. So it's nice to be able to do this in collaboration with a group that can do that kind of work. These are just some known radiopharmaceuticals and the targets that they go to. So if you're doing anything that has to do with substance abuse or Parkinson's disease or sympathetic innervation or Alzheimer's disease, these are known radiotracers that will measure aspects of those diseases that you can use in your project. So as one example, these are some data from here at IU. Imaging using carbon-11 PIB, which is an amyloid imaging tracer, and PBR28, which is an inflammation tracer. So these are AD patients, mild cognitive impairment, and patients that say they're having some cognitive complaints but are sort of clinically normal. And so you can see in the normal patients, we get a little bit of amyloid binding, but by the time you get to full-blown Alzheimer's, you see a lot of red and green. This is the higher binding of the PIB. Same thing with the inflammation. A normal person has a certain amount of the targets, the inflammatory markers in the brain all the time, but that increases with higher amounts of amyloid and particularly tau deposition. The interesting things are that to me is that the inflammation is actually a different spot than the amyloid is in these images. A project I worked on at University of Michigan a very long time ago was looking at cholinesterase activity as a marker of norepinephrine neurotransmission in Alzheimer's disease. And we developed these radiotracers that are actually enzyme substrates. So this is a neutral compound that is an acetylcholine analog that can readily cross the blood-brain barrier by passive diffusion. But once it gets into the brain, can interact with acetylcholinesterase in the nerve terminals of norepinephrine neurons and get metabolized into this more polar compound that then gets stuck in the nerve tissue. So this is what they call a metabolic trapping agent. The higher amount of norepinephrine neurons, cholinergic neurons, the higher the trapping of the radiotracer. So this got to the point of actually doing clinical human studies with it. This is something I started as a postdoc, but we actually got it into patients, which is one of the really cool things about radiochemistry. When you're doing development, it's not a 20-year process, it's maybe a year or two process, comes from bench to bedside, which is great. I put this up just as an example to show you that, first of all, I need to tell you, these aren't actually PET images, these are parametric images. So these are 3D Z-score maps of the difference between a control group and an Alzheimer's group. So in the first line here that says no dose, this is just the degree of difference in cholinesterase activity between Alzheimer's patients and control patients. It looks like it's hotter, but it's actually a big decrease, basically, in cholinesterase activity. The second two are giving what was then a common Alzheimer's drug, donepezil, which is a cholinesterase inhibitor. So we see big differences between controls and treateds in these drug treatment study. The kind of upshot of this paper was that there really wasn't much difference in terms of enzyme activity or enzyme occupancy between a 5 milligram and a 10 milligram dose of donepezil. And 10 milligram is what they were giving people, and they're starting to get some side effects from it. So this, again, is one of the things you can do, these sort of drug titration studies with indirect measurement. I think maybe in the interest of time, I might skip this one. So let me get to the end of this guy real quick. But so you can kind of see what you can do as far as indirect measurements. You're not directly measuring your drug, you're measuring aspects of the disease or aspects of the drug mechanism to get those measurements. And you don't have to redesign the radiotracer every time you want to do a study, if you want to test a new drug. And then the thing that a lot of clinical nuclear medicine is doing now is basically basic metabolic imaging of drug effects or of disease processes. So in this case, you're using a well-known radiopharmaceutical that's just for a general metabolic process like blood flow, glucose metabolism, protein synthesis, things like that, that isn't specific to any one disease, but you can characterize how your drugs are working in relation to those parameters, or you can characterize diseases in terms of, do they have a high glycolytic rate or a low glycolytic rate, and how does that affect the prognosis of the patient. Usually these are, again, things that we're already making here because they're pretty common, or that PET Pharmaceuticals will sell you because they're commercially available. Sometimes they're implementation of published agents. And again, this is sort of the cheaper way to go if you don't need very specific information about what your therapy is doing or what your disease is doing. And almost all of these are basically either commercially available or as fee for service through our radiochemistry, through the In-Vivo Imaging Core. So another list of examples of this kind of radiotracer. We've got FDG, which is the main clinical radiotracer that if you know anybody who's had a PET scan in the hospital, what they've had is an FDG scan. And then all these other radiotracers that are specific for general metabolic processes and can make measurements of those very easily using the image. So one example, this is a O15 water brain activation study. So this is looking at differences in blood flow. Again, these are Z-scores, not actual PET images. Differences in blood flow in different areas of the brain in a patient that they've given ecstasy to. So the point of it was to show that basically the areas that are activated are the ones associated with heightened mood and extroversion, and then decreases in things that had to do with perception alteration and making these patients lower inhibition. But this is all measured just by simple blood flow measurements. So you give the O15 water, remember 2-minute half-life on this, right? So I can inject my patient with water, do the scan, inject them with the ecstasy, wait a few minutes, do a second scan, and do another water injection, do another scan, and then take the difference between those two scans in an individual patient. And it gives me these activation scores. This is just a simple example of a tumor mouse. This is a nude mouse with a subcutaneous, I believe this is a squamous cell carcinoma. And this is the FDG image of that patient. Your brain uses a lot of glucose for energy, so the brain of the mouse is lighting up pretty good. If I had taken a slice that was through the heart, you could see the heart, but this is kind of the tip of the heart right here. The heart also uses glucose for energy, so those show up in all FDG scans. It mostly clears through the kidneys and the bladder, so that's why those are hot. And then this is the tumor right here on the flank of this mouth. And this, again, this is mainly used for clinical and for drug testing for new therapeutics. An interesting radiotracer is C11 acetate, because acetate is central to so many different metabolic processes. You can actually use one tracer for multiple different things. If I'm looking at oxidative metabolism, so I have a tissue like the heart that is very oxidatively active, I can see the accumulation of my C11 acetate in the cardiac muscle. So these are heart images of a normal and a heart with dilated cardiomyopathy. But you can see basically the blood flow and the viability of the tissue around the left ventricle. If I'm in a tissue that's a rapidly proliferating tissue like tumor tissues, then most of the acetate as acetyl-CoA gets shuttled into the fatty acid pool, and I'm looking at membrane lipid biosynthesis. So in that case, if we look at this patient with kidney cancer, these are transverse images. So these are like slicing through the patient like you're slicing a piece of bread. So these are slices through the kidneys right here. And this big black spot that shows up further down in the scan is a renal cell carcinoma that this patient had. So the cancer is taking up higher than the kidneys. So the kidneys, that's the oxidative metabolism you're seeing, and the tumor is the lipid biosynthesis. And if you look at the kinetics of those two spots, they're completely different. So this dotted line is the kidney, where you see rapid infusion and then rapid clearance because it's being metabolized into CO2. The tumor is slower to accumulate, but it stays around a little bit longer, kind of plateaus a little bit. This isn't a particularly good example, but the tumors will typically just plateau out with acetate over time. Another of these metabolic trapping agents are the amino acid radiotracers. There's a number of them. We made C11 methionine at St. Jude. We're making fluorine 18 FET here, and these are being taken into tumor cells by the large neutral amino acid transporter, and then trapped there in one way or another through normal metabolic processes. So this is some old C11 methionine data from St. Jude, and you can see the vast difference in normal distribution between C11 methionine and fluorine 18 FDG in these patients. Again, the FDG shows the hot brain, which becomes the main use for the amino acid radiotracers is for brain tumor imaging, because this is the normal background of the brain in a patient. As I said, we're using the FET, the 4-alpha-tyrosine here at IU. That was one that was sort of developed as an alternative for C11. Again, because of that 20-minute half-life, you can't really commercialize something that you have to make on site. So the companies that sell radiotracers want everything labeled with fluorine 18 or longer. So the FET works pretty much the same, but you're actually able to get a little more kinetic data out of it because the FET actually does clear from the tumor after a while. And this is out of a paper from Dunkel et al. A few years ago that showed a MS patient so basically no tumor, right? And I don't know if you can see the scale in this, but the numbers are only go up to about 2. And this is a standardized uptake value. So it's essentially fold above background value is what it is. But you can see it only goes up to about 2, and it's a really slow accumulation in the brain because there's no real tumor here. This is a full-blown recurrent glioblastoma that's growing really rapidly. So now my accumulation in the tumor is shooting up pretty quickly. It's going up to 4 SUV and staying there with nothing coming back out. This is a sort of middle of the road astrocytoma, so it's not quite as rapidly metabolizing. You still get that big influx of FDG into the tumor, but it eventually clears back out because it doesn't get metabolized quickly enough. So these 3 different patterns are used clinically to diagnose brain tumors in the patients over at IU Health. So this is my team at BRTC. I'll stop here so that you guys can have a couple of minutes for questions. These are the folks that are in my team, including Dr. Green, who's not in the picture. We have a lot of other collaborators in radiological sciences, in particular, Elizabeth Butch is a PhD biochemist who has her own sort of departmental level shared resource for biotesting, doing a lot of these dissection studies and in vitro studies for basic biochemical assays and things for us. So we do a lot of collaborative studies, as do other investigators in radiology. And then just to give credit to the folks at St. Jude that I pulled the data from, and the folks that I collaborated with on some data that I didn't show you actually, and of course the funding, including the most recent CDSI core equipment grant that funded our new radiochemistry modules. So let me stop there and thank you all for your attention. These Zooms are always hard. I'm not sure if everybody's still awake, but I'm happy to answer any questions if you have them. Thanks.
Hi, this is Rafael Polidoro. So I would like to know on the part of IACUC radioactive training, like if we want to collaborate with you, like I have a specific peptide I'm using as a drug, how much of this, because this makes things a little bit slower, right? And until we can start.
So, so Dr. Butch, who I just mentioned, is in the process of writing a blanket protocol for all of the sort of non-imaging pieces of the biological evaluation. So she will have an IACUC protocol, and the way it's set up is that investigators are going to transfer their animal models to her protocol, and we'll do all the radioactive work on her protocol that's already approved to do the radioactivity. So nobody in your lab would necessarily need to learn how to use radioactivity. They're welcome to come and learn if they want. But it makes it a little easier. The only catch there is, then it can't be some model that we would need to then do, therapeutic interventions and stuff on. It has to be like you're done with it and you're just giving it to us and we're going to do the imaging or the dissection and that's it. So that's sort of the break point between the 2 protocols. But that's how I've done it in other institutions, and that's how Dr. Wu also has her own protocol for the imaging, which works pretty much the same way. You just transfer your animals to her protocol, and then she does the imaging on her license, her approvals.
Well, if nobody has other questions, I have more. So there is this genomic core that also does peptide synthesis in the MS building. Do you guys work with them to add isotopes, for example, on the peptide? Because I'm synthesizing peptides with them. Right. So if I want to have it radiolabeled, or you would do the radiolabeling and synthesize yourself.
Right. So I haven't yet, but that's been one of the things that's been on my to-do list actually, because in terms of that antibody project, the biggest detriment of that project is the slow clearance of that full-length antibody. So what I'd really like to do is engineer synthetic peptides that would have a specific reaction site that we can label, so we know exactly where the isotope is. We know it's someplace, so it's not going to mess up binding, and that we can make those as smaller peptides instead of the full-length antibody. And there are definitely published ways to do that. I haven't gotten as far as going to talk to those guys yet, but absolutely, that'd be something I'd be really interested in doing, because I want to do it in my projects. I'd love to get the groundwork, rolling to how that collaboration would work. Absolutely.
Okay, I'll keep on going. So my lab works on gut microbiota, and of course, gut microbiota is a very, very big umbrella compared to the functions the gut can do, regardless of, the genes are more important than the species, but we still talk about it as species. So right. Thinking about metabolites, I think it would be nice because we have, I would say, a few groups of gut microbiota in IU in different departments. I'm in pediatrics, but I know people in the immunology department and biochemistry that probably could start some sort of collaborative consortiums with you guys. Consider what you said about being collaborative and a core to look at metabolites permeability, right? Because yesterday there was a talk, a person was talking about a specific peptide that goes to the brain in vivo that absolutely could be used to be radiolabeled and be used in your core. And I'm almost sure that the author does not know of your core, and I'll actually email her after telling you. So it could be actually a bigger consortium, because gut microbiota and imaging, especially blood-brain barrier permeability, if you already have something that does not cross, that would be really cool to inject that and inject drugs to reverse cerebral malaria. That's my motto, for example.
Right.
They're really important.
Yeah, there's been quite a bit of studies. I haven't done any personally, but there's been quite a few studies looking at specifically at blood-brain barrier permeability using the imaging to make those measurements, and like PGP inhibitors and things like that, to modify that process. Yeah, I mean, if we could establish a sort of standard, if you will, way to label the peptides, so that no matter what peptide we got, if we could get them to engineer into it like one specific amino acid that has a reactive group on that we can get a hold of. And there's again literature on that's not trivial, but it's not, it's not a terrible thing to do. That we can make it sort of standardized, then we could develop a standard radiochemistry process that would work for whatever peptide we wanted to use for it. So yeah, I would absolutely be interested in talking to you more about that. I think that'd be a great opportunity.
Yep, thank you.
Dr. Snyder, I have one question. Thank you for your lecture. It was really impressive, and it seems to be very promising for collaborations. I'm from Purdue, and I'm veterinarian. I work with cardiology. My concern is, in case I want to investigate potential antiarrhythmics in the heart or cardiac metabolism, since the heart will be a moving organ compared to brain or a tumor, would that be a concern in terms of acquiring imaging? Should we do a CT-gated image or a respiratory-gated imaging? What would be the bottleneck for a study to evaluate the heart itself?
Yeah, so the first part of that is sort of a Yu-Chen question, but a physics question. But yes, there are standard methods of respiratory and cardiac gating that allow you to get really good heart images. I didn't put any of them up today, but there are I've seen some really nice cardiac images with both neuronal tracers and with just perfusion tracers. So that's definitely something you can do, not a problem. And most of those routines are like sold as packages with the scanner, right? So I believe, Yu-Chen, you can correct me if I'm wrong, but I believe that our PET-MR has a cardiac respiratory gating software package that goes with that thing, and all the patient scanners are like that. All the clinical scanners have those gating packages. In terms of bottleneck, the biggest bottleneck is the transport thing, and with working with Purdue. I have another collaborator at Purdue that we've been sort of trying to sort that out, because he has just a mouse model that we're trying to figure out what's the more expedient way to do it. Do we make a radiotracer and ship it to him to do the imaging, or does he send his mice down here to do the imaging on our side? As long as it's a radiotracer that's not like N13 ammonia that only has a 10-minute half-life, we can make like fluorine-18 tracers, or anything with a longer half-life than that can be produced here and shipped to Purdue. We have a specific courier who has a radioactive materials license that we send stuff across campus all the time, and they ship things for PetNet as far as Kentucky. So it's not an issue. They come to Lafayette, a couple times a week. We just have to do scheduling on it. It's a small extra cost, but it's not huge. It's like maybe an extra $100 per trip. And if we were going to use something like an F18 tracer, if it was possible to kind of stack up your studies, so where you're doing not just one animal at a time, but maybe you're doing 2 or 3 in a day, you would have one radiotracer cost, one transport cost, and then you could do 3 scans of 3 different animals.
Great. That's impressive. Thank you very much.
Sure, not a problem. Anything else? Well, I really appreciate everybody's interest. Feel free to email me with additional questions, or if you want to connect on anything, I'm here to help. That's what my job is to facilitate these kind of collaborations. So let me know what I can do.